Hi, good morning, everybody, or good afternoon, depending on where you are. I'm Julie Wolf with the American Society for Microbiology, talking to you today about what's hot in the microbial sciences. Microbial Minutes aims to summarize some of the hottest research that has been published in the last couple of weeks. And it's actually only been 2 weeks. We haven't had a delay as we did for several sessions before that. So if you're live, welcome. Feel free to ask a question in the chat box and we will answer it as the presentation goes on. Otherwise, if you're listening later on, feel free to leave a comment if you have a question. All right. I was just at headquarters and would like to say thanks for the great swag promoting ASM journals. All right, now we're going to get started with our presentation. For this, I'm going to share my screen. Seem to be having some technical difficulties. Can I have confirmation from anyone that you can see my screen? Ray, do you see the screen that I'm sharing? I just want to make sure because on my machine it doesn't look like that.
Yeah, I can see your screen. Just make it
Okay.
full size or start the PowerPoint.
Yep. No, okay. Just wanted to confirm. Cool. All right, guys. So what important things have happened in the microbial sciences in the last 2 weeks of October? We're going to talk about a number of things today, with the take-home messages being that measles is on the rise in the US due to lower vaccination rates, that there is a new type of antibody called a tri-specific broadly neutralizing antibody that may work against HIV and other infections. We'll talk about how the immune system works together to stop viral infections. We'll then discuss a little bit of microbiome data, both from a new human microbiome project data report that tells that there are now 3 times as many identified bacterial genes. And finally, we're going to talk a little bit about the soil microbiome and how rising soil temperatures may release more carbon due to soil microbiome activities. All right, we're going to jump right in with a paper from, and here is a list of the papers. We're going to talk about papers from JAMA Pediatrics, Science, Cell, and Nature this week. And so the first one, the JAMA Pediatrics paper, is titled Incidence of Measles in the United States from 2001 to 2015. And as I said at the beginning, the take-home message is that measles is on the rise in the US due to lower vaccination rates. So measles is caused by the measles virus. This is an RNA virus that is extremely contagious. According to the CDC, 9 out of every 10 people that interact with an infected person will contract measles. And this is a disease that is on the list of US nationally notifiable diseases, meaning that if a healthcare worker diagnoses measles in somebody, they are required by law to report that disease to a federal agency where they can keep a list in order to track where within the United States measles is being passed. Now, measles as an endemic disease was eliminated from the United States in 2000. Being endemic means having a transmission chain going from person to person that lasts over 12 months. However, there is still many different incidences of imported disease where people will either travel outside of the US, perhaps on vacation, and interact unknowingly with somebody who has measles and bring it back to the country. Or it could be brought in with people who are from other countries where perhaps measles is still endemic. And when this disease is imported into an area of vulnerability, such as in a susceptible population, that can lead to an outbreak. Now, those areas of vulnerability within the United States are increasing because the parental refusal of vaccinations is increasing. This has been a movement that is well documented and discussed in many different venues, and really arose, let's say, about 20 years ago, thanks to a now very well debunked link to autism, tying in measles vaccine and the preservatives within the measles vaccine to autism. And let me repeat again, this was a study done by Andrew Wakefield, who has both been debunked and had his medical license removed. And there is also a movement that has been kind of spurred from this particular debunked link, where parents are worried about how much immunity is being introduced into a child at once, where parents will change the vaccination schedule because they don't want too many antigens to be exposed, their child to be exposed to too many antigens at once. And let me underscore that there's no scientific evidence for any of these anti-vaccines attitudes, and that the schedule of vaccines as recommended by American Pediatric Association and other medical associations are safe and in the best interests of both the child who is being vaccinated and the population. Measles is given as part of the MMR vaccine. And what this group here wanted to look at is, all right, what is the incidence of measles within the United States based on these nationally notifiable disease lists from 2001 to 2015. And what they found is 1,789 measles cases that have been reported with a median age of 15 years. So that's the age that was most frequently found, but the range was really from very, very young to very, very old. The vast majority of these measles cases were in unvaccinated patients. So those are people who had not received that MMR vaccine. 535 of those cases were imported, and the rest were acquired within the United States. So you can see if we look at this column from table 2 in the paper, that there are the number of imported measles cases, and that has more or less stayed the same within double digits from 2001 to 2015, as listed here in the year. But the number of measles cases that are affected by these imported measles cases has been steadily increasing, staying in the double digits for many years, but then with a big increase into triple digits starting around 2008. And this is due, according to the authors, to the decrease in measles vaccination. This was a story that was shared widely, both on social media and found also in the mainstream news. So here we have Washington Post science editor Laura Helmuth sharing the story. I believe we'll have a link after the presentation is over that you can find to read some of these stories yourself. This is also shared in Time magazine, and they had this lovely graphic that you could look at where those measles cases had been occurring. You could either look in a graphical representation of a map here, where you can see that the highest incidence of measles cases occur along the West Coast here and the East Coast. It's a little increase in Ohio as well. And you could also rearrange this looking at the states in order of incidence. So you would have Washington, California, Ohio, and New York at the forefront, and then those with second most being Illinois, Indiana, Pennsylvania, Massachusetts, and Texas, and Hawaii, etc., and decreasing. So you can kind of play with the data on your own. Interestingly, this is also found in WebMD, so that people who are searching for information about measles in this website, where many people will come to self-diagnose, kind of compare symptoms with what diseases they might represent, will find this article. And I think that that's great for science communications. Here they interviewed the lead researcher, Nakia Clemens, and the researcher herself said that the findings suggest that being unvaccinated rather than failure of vaccine is the main driver of measles spread. Measles is still commonly transmitted in many parts of the world. Unvaccinated U.S. residents returning from overseas travels and foreign visitors to the U.S. may develop measles and expose people in the United States. So hopefully these types of numbers will lead to an increase in vaccine rates. We will only see and hear back in Microbial Minutes in the future. Moving on to the next report, we're going to discuss a new type of therapy for HIV. The title for the paper is called Trispecific Broadly Neutralizing HIV Antibodies Mediate Potent SHIV Protection in Macaques. And so the take-home message from this is that trispecific broadly neutralizing antibodies may work against HIV and other infections. We're going to be talking about HIV, which remains a major killer and one of the top 3 infectious diseases that are causes of death around the world. However, the disease that we will be discussing that is being tested is SHIV, and that's because we're using a macaque primate model, and macaques do not get HIV, they contract SIV, simian immunodeficiency virus. And so there's been a hybrid version of this virus made, SHIV, which maintains a lot of the characteristics of HIV, but is also able to be used in primate models for testing things such as new treatments. We've discussed previously in different sessions how broadly neutralizing antibodies are the goal. So HIV has a very high infection rate, and an antibody that will bind to one of the, I'm sorry, not high infection, high mutation rate. So the areas of the virus that interact with the host cell can actually change in their sequence a little bit. And an antibody that acts against one strain of virus, if that virus mutates in a way that prevents that interaction, then that antibody would no longer be effective against that second strain. And so the goal is to define and use some of these broadly neutralizing antibodies that largely act against areas that are constrained from mutation. So they can't mutate or else they will result in a non-infectious virion. And so there are actually a fairly large library of, let's say, several dozen well-characterized, broadly neutralizing antibodies against HIV. And those are what the researchers here are going to work with. Here they're using an antibody as the basis for a molecule that can interact with several different areas of the HIV virion. An antibody, a normal natural antibody, such as those produced by our B cells, has a constant region here, and this is kind of what helps direct the antibody and help other immune cells recognize what they should do if they detect that antibody. And the antibody can then interact with its variable regions, and the variable regions are these two regions here. Now, normally these are the same. They can a single antibody can interact with its variable regions with two molecules that have the same identity, so two antigens that are the same. Bispecific antibodies are those in which one of these two regions has been genetically engineered. This is not something that occurs naturally, but genetically engineered so that one antibody can interact with two distinct antigens. And that's what a bispecific antibody is. And that seemed like the limit of how many different specificities one could make on an antibody, because there are only naturally two variable regions that connect to this constant region. However, what this research group did was to add a third a variable region, which they added with some connector linker regions here, so that this region here, this variable region, second variable region, would still be able to interact with its designated antigen. And this variable region here would then be able to interact with the third interactive region of HIV. And this first interactive region would not have any difficulty interacting with its designated antigen. And so the idea is that by using three broadly neutralizing antibody regions, the researchers would be able to inhibit HIV more specifically and with better efficacy than a single broadly neutralizing antibody. And so how did they test this? They did a lot of, well, first they had to decide which broadly neutralizing antibodies they wanted to use. And so they ended up using those different antigens or sites within the HIV virion that are required for HIV to be internalized into its host cell. HIV infects CD4 T cells, and so one of the antibodies interacts with the CD4 binding site, the receptor for viral attachment. One of those variable regions interacts with the membrane proximal external region, MPER, and one of them interacts with the V1, V2 glycan site. All of these are on the HIV-1 envelope, so they will hopefully, by working together, inhibit HIV, or in this case, SHIV interaction with host cells. And they tested this by looking at the broadly neutralizing antibodies in singular, as you can see here. Here are two of the three which they had tested, and looking at the activity of these antibodies against two distinct SHIV strains, as you can see here, 1 and 2. And in this case, neither of these singly broadly neutralizing antibodies was able to inhibit infection very well. So this is the titer of antibody that is necessary in order to decrease infection, I believe. However, when they had this tri-specific broadly neutralizing antibody, it was able to inhibit both strains of HIV. And this is in an in vitro system. They then went into the non-human primate model and again tested a single broadly neutralizing antibody, either this VRC01 or PGDM 1400, against that tri-specific antibody that they had engineered 3 different antibody variable regions together. Now, in these different graphs, each different shape represents an individual non-human primate, and we're looking at the viral load within those animals over time after first the antibody is given, and then the animals are infected. Hopefully that antibody is going to be able to inhibit the virus getting inside of the host cells. And you can see that there are some animals that are able to prevent, in which this antibody does prevent infection. However, many of the animals still develop fulminant disease with virus replicating and increasing the number of viral genome copies. Only in the case of this tri-specific antibody are all of the animals protected. No detection of viral copies was made in any of these. And so this is quite an exciting finding because it was a single dose of this tri-specific antibody that was able to maintain all animals at undetectable viral levels. And so this was shared, of course, quite broadly in many different outlets. This was a work that took the word goodness, what's the word I'm looking for? This is a collaborative effort, excuse me, from both the private sector. So Sanofi is a pharmaceutical company that had been working on this, as well as academic and government sectors. So people coming from the NIH, we're all working together toward a similar goal of generating this anti-HIV molecule. So this was shared by people in all of those sectors. You can see here that folks from Sanofi were quite excited at the idea of generating these molecules. This was shared in the nonprofit world. So IAVI is International AIDS Vaccine Institute, and they were also quite pleased to see this very good result. In the independent, Gary Nabel, one of the senior scientists here, emphasized how effective this particular type of antibody is, saying that these tri-specific antibodies are more potent and have greater breadth than any single naturally occurring antibody that's been discovered. We're getting 99% coverage and getting coverage at very low concentrations of the antibody. This is important because there are still over 36 million people worldwide living with HIV, and many don't know it. So people are susceptible even if they are not aware that their partner may have this disease. And finally, this was also shared by medical professionals during ID Week 2017. You can see here is a slide shared by somebody who's attending a talk discussing this very data. So this is quite exciting. And one can imagine that not only will this be effective against HIV, but could also be effective against other very hard to fight pathogens such as malaria or tuberculosis. The causative agents of those might also be able to be inhibited by some sort of tri-specific antibody. Moving on, we're going to talk just very quickly about this paper that was in Cell. This is titled Organism Level Analysis of Vaccination Reveals Network of Protection Across Tissues. And the take-home message here is that the immune system works together to stop viral infection, which may be something that we know, but there is still quite a bit for researchers and scientists to understand about how the immune system works. Their introduction starts with no metazoan cell or organ is an island, which I think is a great opening because many times in an animal system, people will look at how the immunity has developed within the spleen or within the thymus, but not necessarily looking at all different organ systems and how these organ systems interact. And so that is exactly the question that these researchers wanted to ask. How do cells and organs coordinate actions at the organismal scale to prevent infection? And what immune pathways are activated and when throughout the body during local or systemic infection? So to study this, they looked at several different types of immune responses. They looked at a vaccination response using a mouse pox virus. that is attenuated in its virulence, and they injected this subcutaneously as one might for a vaccination. They looked at a lethal immune response where the animals were infected intranasally, as natural infection may occur, with a virulent strain of mouse pox virus. And then they looked at protective infection, where they first gave the animals this subcutaneous injection, similar to a vaccination, and then infected them intranasally. They used a number of different techniques, generating a whole lot of data. They used whole mount tissue imaging, so there are some very compelling images that they use as their figures. They used whole tissue gene expression, looking at how the different genes were activated over time in different organ systems. And because this is a cell paper, they also include a graphical abstract that summarizes some of the data that they found very nicely. So here you can see they're injecting the animal, looking at organism-wide analysis, both by looking at the gene activation across the whole body, taking different organs at different times to see what genes may be activated and what may be activating them, as well as looking at tissue resident memory T cells. So those are T cells that have a T cell receptor specific for this particular virus, and looking at where they are and when after and before viral challenge. So this paper generated a lot of data. We're not going to go over every piece of data because if you're interested, you can read the paper for yourself. They did observe immune signal propagation across organs with both of these infection types, and they observed two major interorgan mechanisms that help to protect the host from systemic viral pathogenesis. So kind of the systems that would be in place to stop an infection from becoming a deadly disease. One of those was the type 1 interferons. So interferon is an antiviral molecule that can act to move throughout the circulatory systems and alert different organ systems that there is a viral attack underway, activating some of the antiviral responses in nearby organs. And they also looked at tissue resident memory T cells, so T cells that would be exposed upon that subcutaneous vaccination route to the attenuated version of the virus, develop specificity in their receptors for that type of antigen, and then be reactivated. And so here we are looking at some FACS plots, looking at where these different T cells, the memory T cells, are within the body 3 weeks after vaccination. You can see, even though the vaccination or the virus was put in a subcutaneous infection, that there's been development of T cells. So these are CD8 T cells. Those are the T cells that are most likely to attack virally infected cells that are specific for this B8R epitope on the vaccinia virus that they were using. And you can see that there is a population of these resident memory T cells that are inactive, though they're selecting for different markers, that reside in the lung, the liver, the spleen, and the lymph nodes, in this case looking at the inguinal lymph nodes. When they then, there we go. They then look at the lung response in some of these T cell populations in animals that have been vaccinated and those that haven't. And you can see within that lung response that that same population now being activated develops a larger population than animals that have not been exposed to the virus. And so this wasn't something that was picked up in the mainstream media, but because of the very large data set, I think that scientists are quite excited about this, and it will serve as a way for new hypotheses to be generated, looking at the activation of immune systems, different systems of the immune system over time and in various types of immune responses. And that's why I thought this was something worthy of discussing, because this paper will very likely be cited many times going forward. All right, we're going to change gears and talk about microbiome for a little bit. So this is another paper that generated a whole mess of data. And so this is from Nature, and it is entitled Strains, Functions, and Dynamics in the Expanded Human Microbiome Project. So the take-home message that I wrote here is new human microbiome project data triples the number of identified bacterial genes. But really, there are all types of different sub-takeaway points that could be included here as well. There are a number of different findings, and we'll talk about just some of them. But of course, you can always go to the paper yourself in order to read and find what some of the cool data is. So what is the Human Microbiome Project? For those of you that don't know, the NIH has been sponsoring a decades-long now project that aims to characterize the microbiomes of diverse cohorts of healthy people. So because of the difference in genetics, like people are not mice that are used in a lab. We all have different genetic systems and we have different diets, and those things will influence the microbiomes that are found both inside of us in our gut, as well as on our skin or in various other areas of the body, in the lung, etc. And so this is a new report with samples that have been looked at by whole metagenome sequences. And taking this metagenome means that they are not amplifying the nucleic acids before looking at what the sequence of those nucleic acids is. And that's in order to help them to lose any bias that amplification would have. Sometimes if you amplify those nucleic acids, you'll end up with a different proportion of certain sequences that are able to be amplified more efficiently than others, and you can get artifacts in your data from that. So they have 1,600 samples here, including 265 sampled individuals from various body sites. And the goal of this project is to find some of those key properties of the human microbiome that remain to be characterized in healthy cohorts. In addition to microbiome contributions to disease. So in this particular study, we're looking at healthy individuals. We've previously spoken about sick individuals or the role that the microbiome may play in certain types of disease. Last week we talked about, or 2 weeks ago, we talked about multiple sclerosis. In the last couple of weeks, there have been several very interesting studies that I thought might be candidates for our microbial minutes, tying in the microbiome with diabetes or looking at the results of different types of birth, comparing vaginal birth with C-sections. However, there's just so much left unknown about the human microbiome itself and what it looks like in a healthy situation that sometimes it's important to remember that we're at the very beginning of this field, and learning what an unhealthy microbiome looks like is something that we can't really define very well just yet. And so to emphasize this, I want to talk about one of the findings that they had, which distinguished temporally stable subspecies that are associated with particular body sites. And here I'm going to quote directly from the paper with the bolded text there is bolded by me. Here the authors write that composition of the gut community, Bacteroidetes species in particular, are highly personalized compared to other sites. Bacteroidetes firmicutes balance may not be a defining attribute of an individual's gut microbiome. One carries personal equilibrium among Bacteroidetes with a group of phylogenetically diverse, temporally diverse variable firmicutes fluctuating atop this core. And I apologize if I pronounce that different than how you're accustomed to hearing. I've heard Bacteroidetes, and I actually like Firmicutes to keep within the same type of pronunciation, but more commonly I hear Bacteroidetes and Firmicutes. Of course, these are some of the words that we often read and don't always say out loud, so if you prefer a different pronunciation, just let me know. But I wanted to emphasize this because there are many papers that talk about dysbiosis within the gut, using Bacteroidetes and Firmicutes as a marker of health or unhealth, respectively. And what this is suggesting is that there isn't one correct ratio of these different bacterial groups, but that correct and healthy ratio may be specific for each individual. And so to say that the ratio of these two different bacterial groups is what is causing disease is really hard to do at the individual level unless you know what that ratio looked like in that same individual when one was healthy. I'm showing you here a supplementary figure to the paper itself, where they look at stability of these different Bacteroidetes groups, which are in red, and show that the firmicutes are more along the time varying bacterial groups, whereas the Bacteroidetes seem to be a fairly stable group within each individual. They also did a number of other interesting studies, sub-studies within this report, where they resolved the co-occurrence patterns, looking at the bacterial abundances, as well as those of several archaea, eukaryotes, and viruses. So that's another place where using the metagenomics comes into play. I didn't note whether or not they looked at RNA viruses, but by just looking at what DNA is present, one will be able to look at the genes from viruses and eukaryotes, such as fungi or protozoans that might be inside of the gut, as well as, of course, archaea, which are also present in the human gut and other places along the microbiome. And they also use those genes that they found to identify new functional pathways that are required for the microbial colonization of the human body, which is a different type of niche than many other niches, such as environmental niches. And this helped them to differentiate pathways that are enriched for human habitat, which are different from those that are universal for microbial life writ large. So this was something that was written up in many different mainstream news places. Here I'm showing genetic and engineering news, where the senior principal investigator Owen White says that this new data really expands our appreciation for the fingerprint created by microorganisms that make up each human's microbiome. And the take-home message from a lot of these different write-ups was, hey, there's a lot of information here. And so I think just like the cell paper, this is going to serve to be a hypothesis formulating paper where people will delve into the data and be able to make new hypotheses that they can then test going forward. This paper also created a little bit of controversy when one user, so Jonathan Eisen, is a large proponent of open access papers, particularly those that are funded by by U.S. taxpayers, and he was calling out Nature, who was advertising this or promoting this paper, by saying that not everybody would be able to have access to this particular research, which is a shame because Eisen, working at an academic institution, likely has institutional access, but many taxpayers do not have access. There was also an interesting discussion among scientists, which I've highlighted here on the right-hand side, talking about that raw data. So the raw data from these types of sequencing studies is often gigabytes worth of information, and storage, it can be quite difficult. And so, like many other data scientists, this will have to be stored on a different server, and here it's stored on an Amazon server. Amazon, of course, storing a lot of digital data as well as selling things online. And apparently this is data that is available for people familiar with bioinformatics to look at and play with some of that data. So if you have any interest in looking at what the raw data looks like, you can follow this link here, where the first tweet says data available on AWS, but also a debate arising about who should pay for that, whether that storage, whether it be the funding agency, the NIH, or the researchers. And so that is something that I don't think was very well resolved, but I thought it interesting that this data is available, even if you don't necessarily have data or access to the paper itself. Now, I'll give you a hint about finding some of these papers that you may not have institutional access or an individual subscription to. If you go to something like the genetic engineering news site, you can often click through to the hyperlinks within those mainstream news sites, and it will take you to the original report. And sometimes publishing companies will provide an open access link that is available only by a certain link. So in this case, only through the genetic engineering news. So I tested that just with this one particular report and was able to access the report itself through genetic engineering news. Also, sometimes through Twitter, different publishing agencies will make an open access link or an online only accessible version. So there are ways to get around not having access sometimes if you just click around and find and experiment with different links. All right, finally, we're going to talk about our last report, which is a I believe this was also a science paper, and this is entitled Long-Term Pattern and Magnitude of Soil Carbon Feedback to the Climate System in a Warming World. Take-home message from this being that rising soil temperatures may release more carbon, which may sound like something that we already know, but actually is very hard to test. So what the researchers here want to ask is, how does global cycling of carbon and carbon cycle feedbacks affect climate change? And how does climate change affect the global cycling of carbon? Carbon is released through respiration as CO2 by humans. Most people don't consider that to be a major factor in release of carbon, because we are also consuming a lot of carbon. But the microbes that live around the world, both in the ocean and in the soil, can also degrade different nutrients, carbon-containing nutrients such as sugars and lipids and things like that, and release byproduct of CO2. And computer modeling has been, a lot of computer modeling has been done looking at the role of microbes and how this release of carbon can either slow or accelerate warming, with modeling suggesting both, depending on what variables are investigated. And in order to verify the models and to refine them, scientists need actual data. And that's why this is a really cool study, because this study has been going on for 26 years, looking at soil warming as an experiment designed to explore these very feedback issues. So this experiment started in 1991, a long time ago, in central Massachusetts in the Harvard forest. That's what's pictured on the right-hand side here. And in the Harvard forest, the scientists set up several different plots with one of three conditions. Some plots had cables that were run underground that would heat the plots, that particular area of soil, 5 degrees Celsius above whatever the ambient temperature was. Some plots had disturbance control, meaning that they had cables that were run underneath them, so that in case planting those cables disturbed the architecture or the microbial composition or oxygenation or some other unknown variable, that would be controlled for. But there was no power run through those cables, so that there should not be a warming effect. And then there were also undisturbed, or as I wrote here, undisturbed control plots. And what they found were that there were several different phases of response. And here I'm taking not a figure from the primary article, but there was also a commentary that accompanied it. So if you're interested in reading still a relatively broad summary, but perhaps a little less detailed than the report, you can go ahead and find that as well. And what they observed were 4 different phases of the response. Beginning in 1991, they saw a decrease in soil respiration, right? So that's in this section here, going all for the first 10 years or so. And what was happening at this time was that the microbes were using up some of that accessible carbon. So there's different types of carbon that are stored in the ground. Some are in other microbes. That's pretty accessible, because a lot of those things can be, a lot of those biomolecules can be broken down. But some of it is in really hard to access forms, such as in lignin, which is a plant product and something that is very hard to break down. It takes a lot of different types of enzymes to break that down and access that carbon, and then cycle it through metabolic processes to even generate the CO2. And so some of that recalcitrant carbon remained, but most of that accessible carbon was decreasing during this time. And so at this point, they saw increase relative. It started at the beginning with increased respiration relative to the plots that they had seen, but then that steadily decreased. And then there was a period of no significant increase in respiration. So it just kind of held steady. And then as the microbiota of the soil shifted to those types of microbes that can more readily access this inaccessible carbon form, there was another increase that was observed in respiration again. And in the last, say, 4 or so years, there's been a steady decrease, perhaps as that lignin has been utilized. And in fact, the researchers noted that 17% of the carbon from the topsoil layers has been lost over this process. And so this is something that involves a lot of different studies. There's been several publications on this too. Just because it's published now, the experiment will continue to be ongoing, as small samples will not disturb the plots, and they can continue to be maintained at their either ambient or higher temperatures. In addition to looking at the amount of carbon that is there, the type of carbon, they did studies that looked at the microbial composition, looking at the 16S rRNA gene, looking at enzymes that are present, and even in some metagenomic studies. But of course, those are more recent, with that type of technology only being available in the last 10 to 15 years. And so the take-home message from this is quite dire, since it does suggest that increased temperature will lead to more carbon dioxide release, right? And that carbon dioxide, as it's released, will add to the warming effect, the greenhouse effect, trapping in the sun's heat after it passes into the Earth's atmosphere. So this was something that was shared not only by researchers, as you can see here, a lab was able to share this data. It's a long-term experiment. People are very excited about this. This was also shared by nonprofit agencies. So there's a nonprofit Global Carbon Project looking at how carbon cycling is being affected and will affect global warming. And so this was shared. And this article by Chris Mooney is the one that is referenced in the Washington Post by this Twitter user here. And here, not only were the original authors interviewed, stating how dire they found their results to be, but also somebody who was not involved with this particular study, who was questioning a little bit about how the study was run. This particular scientist saying that this study looks at one type of ecosystem and in one single place in the world. And of course, there are different biomes and different ecosystems, and those soils will react differently. However, I will point out that there is no research stating that there is less carbon emissions occurring. And so it's never a bad thing to prepare, given the most dire potential consequences, even if those consequences are maybe a little more stringent or hyped than what they potentially could be, looking at many different ecosystems. I mean, these types of experiments are going on throughout the world. So I do predict that we will talk about these in Microbial Minutes going forward, and that the results will be quite similar. So with that, I'd like to summarize just some of the things that we've talked about today. We talked about the rise in measles due to fewer vaccinations within the US. And we also talked about a new type of antibody that may work against HIV, called a trispecific broadly neutralizing antibody. We looked at how the immune system can work together to stop viral infection. And I did not change the summary here, but the new Human Microbiome Project data looked at a change in the way that we define dysbiosis, as well as revealed a number, 3 times as many genes that are associated with the human microbiome. Finally, we looked at how rising soil temperatures may release more carbon. With that, I'd like to thank you for listening. If you have any comments going forward, would be happy to answer those. So go ahead and leave a comment in YouTube if you found this interesting. I'd like to thank Roy Ortega for helping me to set this up. And we'll talk to you next time in another 2 weeks for Microbial Minutes. All right, thanks everyone.